Hi, this is Miss Litton, and this is lunchtime on Friday, and we are reviewing for Chapter 5, Mitosis, and also cell development. We're going to review that as well. Say hi. Hi. They just have their mouths full, so it's hard. All right, so the things we need to review for mitosis. First thing I would recommend that you review, you can't see, sorry, is the cell cycle. Okay, you want to go over your cell cycle. So you want to remember the bulk of the cells, time is in interphase, all of this is interphase. And we have G1, we have S, and we have G2. What are the key things that happen during G1? Grow. Why do we want to grow? So we don't have to memorize this. Why does it make sense that we go right into growth? They just divided. Exactly. They just finished cytokinesis and the cells are smaller. What does the S stand for? Synthesis. What are we synthesizing? DNA. This is when we are making the sister chromatids. We can't see them as rod shapes yet, but you're actually making the sister chromatids. G2, what are we making a lot of? Proteins. Good. Okay, this next stage right here, this whole stage is referred to as the M stage, but just this region is actually mitosis, and then you have what? Cytokinesis. And cytokinesis overlaps with what stage? Telophase of mitosis. Yeah. So mitosis is specific. It's referring to just the what? Nucleus. It is nuclear division. Cytokinesis is division of the cytoplasm. Good. All right, so make sure you differentiate that. We talked about control of the cell cycle, okay, and there were two things there. Cyclins were like your ticket that promoted whatever process, whether you're going into the S stage for synthesis or mitosis for cellular division. That's your ticket to ride. To check to see if everything is progressing well, those are your checkpoints that we have here, 3 different checkpoints, and we say, oh, did we do DNA replication right? We'll check here in G2 because the previous stage S. We're going to check it here. We did that replication right. We can move on. Are we progressing through the mitotic stages? Okay, maybe drink it instead of doing that. Are we progressing through the mitotic stages well? If so, move forward. If not, gives the cell time to fix it, but if it can't fix it, then what? Then kill it. Exactly. Did we finish mitosis up well? If not, fix it. If not, kill it. So those are our checkpoints. All right. When we say kill it, what's the step called? Apoptosis. Good job. All right. Now, as we move into prophase, the first stage of mitosis, this is when the chromosomes become what? Visible. That process of winding up, they're winding up on those nucleosomes, those histone proteins, to condense so that they become visible. And when they do first become visible, they appear, each chromosome appears as doubled. Why is that? Because it is. Okay. You have 2 copies. One for this cell and one for that cell, right? That you just copy the DNA. It doesn't matter if you're going to go into mitosis or meiosis. You need the copies of that DNA, all right? So if you do mitosis, my arms, my legs, mitosis, all you're going to do is line up all your chromosomes in the middle and separate this sister chromatid from this chromatid. Do you agree with that? And you go from 1 cell to being what? 2 cells. If you're going to do meiosis, I'm going to need, yes, that's exactly what I'm going to need, a homologous pair. And the first division will separate these homologous pairs, and now I have 2 cells. And then the second division will separate the sister chromatids. Okay? Clarifying questions on big ticket items like that. Swedish. Okay, next. We learned the words haploid and diploid, right? So diploid is when you have all the homologous pairs together. Mom gave you a chromosome 8, so did Dad. Mom gave you chromosome 1, so did dad. Those are all your body cells, your somatic cells, full complement of cells. When you go to make your gametes, and the only way you and I can make gametes is through the process of not my arms, my legs, mitosis, but what? Meiosis. When you make gametes, that's when you separate out the homologous pairs from each other. As soon as you do that, they are considered not diploid, but haploid. Exactly. So if I say the 2N number, what is that referring to? 2N. Diploid. Okay? If you just look di means 2N. That's what you're going to remember, right? Haploid haploid sounds like what? Half. Yeah. So only sperm and egg are haploid. So our 2N number is equal to what? 46. So our N number found in our sperm and egg is what? 23. And 2 times N would give me 46, yeah? Okay. Just double checking on that. Next, talked about how plants have meristematic tissues that are always making mitosis. For us, all we have now is really in our bone marrow, is where it's still making more cells. Basics of mitosis, chromosomes become visible, right? In fact, let's go through it. Ready? Let's go through the stages. Let's see. Oh, what's this purple thing right here? Nucleus. What? Nucleus. And what's inside of that that's kind of dark usually right there? Nucleolus. Right? What happens to those things during prophase? Gone, right? All right, the chromosomes, what are these things right here? Centrioles. And those double up, you usually just have just the perpendicular centrioles, which to us look like something from Disneyland like churros, right? You double them up right before mitosis and they move to either end and they are like poles, like on a clothesline, right? And then the microtubules shooting across help push and pull line them up and push them and pull them apart and move those chromosomes around. Those come shooting out and around. And then we have a stage where they all line up, a stage where the sister chromatids separate, and then we form 2 cells. I'm going to go through the song with you right now.
What's the difference between the centriole and the centrosome?
Centrioles sit in the centrosome. Centrioles sit in the centrosome. Plants have centrosomes. They don't have centrioles. Why? I don't know. I don't know, but they don't. They still have what's called an MTOC, microtubule organizing center, where all the microtubules are shooting out of the centrosome, but they just don't have the centrioles. All right, so let's go over mitosis. First, put down your lunch for just a second. That looks really good. First, how many chromosomes are in this cell? 2. 1, 2. Okay, so there's 2 chromosomes in this cell. How many homologous pairs do I have? 1 homologous pair. 1 pair. 2 chromosomes, 1 pair. This is a pair of chromosomes, right? All right. If this one came from mom, this one came from dad. What happens during the S stage of interphase? Okay, is that a homologous pair? No, what are these? Sister chromatids, held together by a centromere. This is still a homologous pair. Just each member of the pair has been doubled up. Why? Because we're getting ready for cell division. How many chromosomes are in this cell right now? 2. Okay, only 2 chromosomes, but each one is doubled up. All right. So what's the whole thing called? Not cellular respiration. Mitosis. What's the first step called? Prophase. The chromosomes become visible. The nuclear envelope and the nucleolus disappear. You could walk around the other way. Okay, that way. Okay, so let's try that again. The chromosomes become visible. The nuclear envelope and the nucleolus disappear, radiating microtubules. They meet in the middle at metaphase, away at anaphase, to nuclei at telophase, cytokinesis. Perfect. Okay, perfect. The cytokinesis happens when we're doing this part. It kind of overlaps on it. When we say away at anaphase, who's going away from each other? Sister chromatids. Perfect. Okay. your chromosome? Yes. Who's that cell? Name that cell. Say my name, say my name. My name is bacteria, and I have a circular chromosome. Okay, how do they divide? By binary fission. That single circular chromosome replicates and then separates into two cells. Do you have a memory of that? Of course you do. All right, let's check and see how you do. Why don't you go in? Is your favorite thing to eat for lunch? Once it loads, once I remove AP. Do the song with your bio buddy right now. See if you remember it. Chromosome? Yeah. While I'm waiting for this to, then I'm going to pause you just for a minute. Okay, we're going to come back and do our answers. Oh, somebody just got into Italian deli. So sorry. Good job. Chromosomes in the middle, metaphase. Proteins are synthesized during G2 of interphase. Good job. Proteins are synthesized. Chromatids are separating away at anaphase, so most of you got that. Division of the cytoplasm, cytokinesis. Membrane breaks down, prophase, the nuclear envelope, nucleolus disappear. Organelles are formed during G1, cell grows. DNA is replicated during S, good job. Nuclear envelope is reforming and you have two cells. So nuclear to nuclei, because I said that, that's why I would put that in the telophase part. Okay. All right, now changing gears, we need to talk about meiosis, and this is where we need to make haploid cells, right? So it is going to be the same in that DNA replicates, but this time we have two divisions. The big thing about meiosis, if all you did was said, you know what, just save us the time, let's just not replicate the DNA, send chromosome 1 here from mom, chromosome 1 here from dad. I could see that argument. The problem is that you would miss out on the opportunity to have a chance at what starts with a V? Variation, yeah. To have variety, to get some new combinations of traits together. So, meiosis, we come back to those homologous pairs that we talked about at the beginning. And during the S stage, they are going to replicate. But now, in the very first stage of meiosis, which is going to be prophase 1, the homologous pairs are going to do synapsis. They're going to snap together. And when they do that, non-sister chromatids, not sister chromatids, but non-sister chromatids can exchange pieces of their DNA with each other, and this increases variety. That happens during prophase 1. Where's our next place we can have variety? Yes?
Metaphase 1?
And metaphase 1, because they line up independently of each other in metaphase 1. This homologous pair lines up independent of the other one. It doesn't say, oh, the girl's on the right, oh, the boy's on the left. It doesn't say that. They can line up independently however you want to line up, so long as you're in the middle. Where's our third area or opportunity for variety? I'm trying to make more people think. Yes? Fertilization. You don't know what sperm is going to hook, that it was randomly made due to crossing over an independent assortment, is going to hook up with what egg who was randomly made over independent assortment and crossing over, they're going to get together. So that's our third area. Great job. So in meiosis 1, the goal, separate the homologous pairs from each other. Now you have two cells. Meiosis 2 happens in two cells. Meiosis 2, you're going to separate out the sister chromatid. But otherwise, you go through those same stages again. Here's prophase 1, crossing over, right? Here's metaphase 1. They meet now they meet in the middle at metaphase. They meet with their dance partner, right? Away at anaphase. This time you separate homologous pairs. Telophase 1 and cytokinesis. Already you can see some opportunities for variety in there, right? New combinations of traits. Okay, then both of those cells are going to undergo meiosis 2. And you're going to have prophase 2 visible, but there's not going to be any crossing over because you've already separated the dance partners from each other, right? You've already separated the homologous pairs. Anaphase 2 and telophase 2 cytokinesis will generate 4 cells as a result. Now, when we talked about why are the puppies different, right? So tell me again, what are the 3 reasons why the puppies are different from each other? Crossing over, which occurs when? Prophase 1. Another independent assortment, which happens during metaphase 1. And fertilization. Right. Good job. All right. Good, good, and good. And then the last thing we talked about in that was how spermatogenesis, the making of sperm, and the making of eggs are different. Because the making of sperm, you will end with 4 sperm cells. But when you make eggs, you do unequal cleavage so that you have one large egg and then the rest are just little polar bodies that will deteriorate. They have the right DNA, but they don't have enough organelles and cytoplasm that they need. So they just break down. Okay, and then here you go for that. And remember, after this, we're going to review chapter 22 real quick, too, because that's the other part of your quiz. I'm going to pause you just for a minute. It just looks like some people opted out of a name or 2. Okay, let's see how we did. Okay, sister chromatids separate. If you're separating the sisters, it's got to be anaphase what? 2. If you're separating the sisters. Got to be anaphase 2. Nuclear membrane rebuilds for the first time, 2 cells total. That's got to be telophase 1 because it's rebuilding for the first time. 2 nuclei at telophase, so we're rebuilding the nuclei the first time, so it's got to be telophase 1. Number 3, crossing over. Where does that occur? Prophase 1. 4 haploid cells at the end of telophase 2. Somebody said prophase 1. 1 cell then. 1. Okay. 5. Single chromosomes are being formed. Nuclear envelope may be breaking down again. Okay. When did nuclear envelope, nucleolus? If we're doing it again, it's got to be prophase 2. If it was the first time, it'd be prophase 1. Number 6. Homologous pairs are separating. Whenever you separate, it's got to be an anaphase. Any kind of lining up in the middle, it's got to be a metaphase. Any nuclear envelope reforming, it's got to be a telophase. You just need to decide which one is it. So here they're saying something separating, and it's the homologous pair, so it's got to be anaphase 1. You're in 1 cell separating. Here you're in 2 cells separating, right? If you're at anaphase 2, you're in 2 cells. 1 cell, meiosis 1, yeah. 7, homologous pairs are in the middle. If your pair is in the middle, how many cells am I in right now? I'm in 1 cell, so it's got to be metaphase 1. Single chromosomes are in the middle. It's a metaphase. They're single. They're not with their partner. They are not with homologous pairs. So that's got to be metaphase 2. All right. That is the end of that chapter. Let's transition into development. Now, for this, I'm going to tell you right now, what I would like you to do in your mind or on your notes is name the stages of development and a key word to describe each stage. See if you can do that right now while I'm opening up the notes. Start with zygote, and then what's the next stage, and the next stage, and the next. You need to know those. Yes.
We don't have a lot of time. Can we do the quiz for it?
I'm not going to share the quiz. Yeah, it's too long of a quiz.
Okay.
Dropbox. Okay. Open file. Notebook honors. Oh. Okay, so let's go really quick on here, okay? I'll just hit the highlights of what you need to get on that. I don't know why I'm having trouble here. Notebook file, sorry. Notebook honors.
Thank you.
Okay, you're welcome, sweetheart. Okay. Key events. Hi, guys. Key events. Know your stages. Know cleavage, morula, blastula, gastrula, nerula. Okay, those are the key stages that occur in each one. Cleavage, you're making more cells rapidly without any growth. You are a morula, you are a what? Solid ball of cells. A blastula, you are a what? Hollow ball of cells because you pumped in salt, water followed. Gastrula, you are a layered ball of cells. Know the 3 layers and what comes from each layer, right? Review that. And then a nerula, you have developed the beginnings of your nervous system. Okay, so make sure to review those portions of it. Let me find, it's this slide right here that you would look out for that. And then the other part I would refer to is about totipotency, that word, and differentiation, how it varies in the cytoplasm, and then that determines the direction that the cell goes in. Review anterior head, posterior tail. Vegetal pole is where the yolk is, and that's the front, and dorsal pole is out the back. Review that part. Review the words morphogenesis and induction, those 2 key words there, and apoptosis. And then I think you should be okay. All right? Have a great rest of your day and weekend, and I will see you Tuesday morning bright and early. Have a piece of toast.